Hello Helsinki, I am Kirmo Vartiovaara and I'm really happy to tell you about our Helsinki Challenge team efforts to make the future a better place. We work here in the San Francisco Bay Area with local hospitals and universities. This young lovely lady here is a patient at the Children's Hospital. We can call her Emily. She is now 6 years old and has a difficult inherited condition that is lethal unless treated with blood transfusions every 3 weeks or so. Emily's disease, beta thalassemia, is the result of genetic failure in the hemoglobin gene. It's noteworthy that the patients are born healthy, since the fetal hemoglobin gene is perfectly normal. Emily and her family are not alone. An estimated 5% of the world population carry some inherited hemoglobin gene mutation. And like Emily's parents, they are healthy, but in a 25% risk of having a seriously sick child. Without treatment, tens of thousands of children die every year, and even with good health care, life is difficult, and treatments cost billions of dollars. These numbers tell us that the challenge to help people with inherited diseases is huge. The positive side of this is that naturally also the benefits would be immense. Furthermore, correcting something like thalassemia would really make a change, since we've never been before able to correct these biological errors. Why are we concentrating to hemoglobin? In addition to the great numbers of patients, there's a good biological reason for this. We all produce millions of new blood cells all the time. That if we can fix the genes in Emily's blood stem cells, the corrected blood cells will rapidly replace the non-functional ones. And that's why blood is a very good tissue to start the therapy of genetic disorders. This kind of stem cell DNA surgery, if you will, uses very recent biotechnology. We have succeeded in editing the insulin gene of stem cells, as can be shown here. If we can modify the stem cell genetic code, and we can, what would we like to do? We could repair Emily's and every family's own specific mutations, but we came up with another idea. We want to make the genetic surgery to reactivate the healthy fetal hemoglobin gene, a useful condition that we sometimes see in the patients. This allows us to use the same intervention for everybody, which is much easier, not only technically, but also from the regulatory perspective. In practice, the methods look something like this. We take the cells from patients, culture and manipulate them in the laboratory, and then we measure the end result. Our team from 4 continents covered the road from the patient to the laboratory and back. We have 2 main sites of action, the Academic Medical Center Helsinki and the Children's Hospital Oakland Research Institute in California. We also have now a valuable mentor, the Red Cross Blood Service, Veripalvelu, with world-class know-how in blood products. It will take the spring 2015 to show that the basic idea works in the laboratory. The techniques can be improved forever, but next summer we hope to be in a situation where some of you in the audience maybe can support us. In the fall 2015, we need to talk to the hospitals, regulatory instances, patient groups, ethical authorities, how to test the approach in patients. We need to get fundraisers involved and are approaching already biotechnology companies in the San Francisco Bay Area.
We are excited about the project, and the whole team is working hard to get results. If you have comments, questions, feedback, please let us know. You can reach us through Skype, email or Twitter. Thank you for your attention. Kiitos mielenkiinnosta.